In this unit, we're going to look at the cardiovascular system and the fluid that moves through it. So, the fluid, of course, is the blood, and that's what this chapter is about. The next chapter is the cardiovascular system. So, I meant to say in this unit, we will cover the blood and the cardiovascular system. So, today we're going to focus a lot on the components, what's in blood. And then next time, if we don't finish that part of it, we'll finish that and then go into some of the processes that occur within blood, like blood clotting and what are blood types, things like that. Um, if we look at this next slide, and you can see, when you go and you have your blood drawn, one of the things they do to that initially is run it through a centrifuge. So that's that little machine you see on the right. And that spins it so fast that anything that's heavy moves to the bottom. And so you can see that in this little test tube. You can see that what's in the bottom is the red part. And that's the red blood cells. Okay, and it makes up about 45% of the total volume of blood. The yellow part is the blood plasma. That's the liquid part of blood. It makes up about 55%. And so if you add 45 and 55, that's 100, isn't it? So where are the white cells and the platelets? They are actually less than 1%. Okay, and they are in what we call this buffy coat. So right there is where the other solid components fall. Um, on this is a drawing of what it might look like under the microscope, and you see a lot of the little red blood cells, and then the white blood cells are these larger ones that actually have a nucleus. We're going to learn a lot about, especially the red blood cells today. White blood cells have a nucleus, red blood cells do not, believe it or not. But they're red because of a pigment molecule that's in them. And because they are red, and because they make up 45% of the volume of blood, it just gives the blood the color of being red. When technically, if you really look at this, the blood plasma doesn't really have much color to it. But there's a lot of dissolved substances in that. And so as we look at the components of blood, we'll also look at what's in the blood plasma. So here you can see the percentages laid out. A medical test that is done on blood very frequently is one called the hematocrit, or packed cell volume, and that tells you the percentage of red cells in whole blood, which is very important because the red cells are the ones that carry oxygen, and so if you don't have enough of those, your cells aren't going to get enough oxygen. So we'll look at that a little bit as we go through the chapter. I like this slide. This sort of breaks down everything that's in blood. In a drop of blood, you have what we call the solid components, or the formed elements, which are the red cells, the white cells, and the platelets. And then the blood plasma, you can see a lot of the components that are in blood plasma. So we'll look at some of these and break these down just later on in the chapter. And then with the white blood cells, do you see there's several different kinds of white blood cells? Okay. So once you have gone through all your notes and gotten through all of the structure of blood, what the composition is, then this is a really good slide to go back to and just talk through, explain each of these. So it's a real good breakdown of what's in a drop of blood. Okay, so if we're going to spend our time today on the solid components, and those are the red cells, which are also called erythrocytes, the white blood cells, which are the leukocytes, and then the platelets, which are called thrombocytes. And platelets are not whole cells. They're solid particles made up of fragments of a cell in your bone marrow that broke up. And but they are considered solid components in that they're not dissolved in the blood plasma like chemicals are. If you were to look at bone marrow, red bone marrow, under the microscope, these are the variety of cells that you would see. Red bone marrow. One of the functions of the skeletal system is to produce your blood, and that occurs in the red bone marrow. We looked at that way back in A&P 1. But there's a lot of different cells in the bone marrow that ultimately give rise to all the cells and the platelets that are found in circulating blood. So here you see red blood cells, and then here's a bunch of little platelets, and then the rest of this would be a variety of white blood cells. Okay. But do you see how all of that comes from that cell right there? And that cell is a stem cell. It's one of those pluripotent stem cells you may have remembered from AMP1. It's partially specialized. It's not totally undifferentiated, it's partially. And but it can give rise to a variety of cell types based on what hormones are influencing it at that time. All right, and so you see that on this slide. Those stem cells, they have a couple of different names. You can call them hemocytoblasts, or a newer name for it is hematopoietic stem cell. Have you ever heard this word from AMP1? Hematopoiesis? Hematopoiesis? What is that a definition of? What is the definition of hematopoiesis? The process that makes blood cells. Good, good. So the name for the formation of blood cells in general is called hematopoiesis, and it occurs in an adult. It occurs in red marrow. And the actual stem cell itself in the red marrow can be called a hemocytoblast or a hematopoietic stem cell. Hemo means blood. Hemo means blood. Cyto means cell. Blast is a cell that makes something. So a hemocytoblast is a cell that makes blood cells. Does that make sense? Remember, fibroblasts make fibers. Osteoblasts make bone. Hemocytoblasts make blood cells. Any kind of blood cell. Okay, so if you look on that slide that breaks it down as to what's in the bone marrow, I'm going to move to that slide. I'd like for you to stay on this one. Oh, right here. Let's see if this eraser will work this time. Okay, so if we take, here's the hemocytoblast. And so under the influence of certain growth hormones and growth factors, it will either divide and somewhat specialize into what we call a myeloid stem cell or a lymphoid stem cell. And once it's that kind of stem cell, then it's influenced by different growth factors or hormones to become a variety of more specialized cells. The lymphoid stem cells give rise to lymphocytes, one of the types of white blood cells. And we'll learn, especially when we get to the lymphatic system, that there's actually two different kinds of lymphocytes. Under the microscope, initially, they look just alike. And in this chapter, we're just going to talk about them as lymphocytes, not the two different kinds. But a lymphoid stem cell gives rise to those. A myeloid stem cell gives rise to everything else, either the red cell, any of the other white blood cells, or platelets. But then, it in turn specializes based on different growth factors. So, and you can see that on this slide right here. You hear sometimes about leukemia. While I'm on this, leukemia is a cancer of cells in the bone marrow. You ever heard? Did you know that? And it can affect the lymphoid cell line, or it can affect the myeloid cell line. There's different types of leukemia. You may have heard those terms associated with some of those different kinds of leukemia. But if a person has that, they're producing too many of, and it usually affects that white blood cell line. They produce too many of a certain type of white blood cell, and they're not normal, so they're not functioning correctly. And because these stem cells are spending so much time making the white blood cells, they don't spend enough time making the red blood cells. So some of the symptoms of leukemia are symptoms of anemia, which is loss of oxygen to your cells, because you're not making enough red blood cells. So that's why when they have a bone marrow transplant, they're getting new initial stem cells in there that are not cancerous, and so they'll divide normally. Okay. So we're going to focus on red blood cells for the next several minutes. And you're going to learn more about a red blood cell than you thought was possible. You can spend 20 or 30 minutes studying just these red blood cells. Okay? The other name for them is an erythrocyte. If I ask you what is the shape of a red blood cell, the shape is described as a biconcave disc, concave on two surfaces, like a donut with a membrane in the middle. That's a biconcave disc. So you can see a picture on the right that shows red blood cells. These are the smallest cells of the body, I believe, except for the human sperm cell. And, oh, I didn't know it flipped over. Sorry. If you look at, you don't have to know the dimensions of it, but look at that. That says 7.5 micrometers or microns. Do y'all have any idea how big a micron is? Okay, there are 1,000 microns in a millimeter. And you know how big a millimeter is. There are 1,000 microns within that tiny little bit of space. So obviously these are tiny cells. Only a couple of microns in diameter, and the height, not diameter, the height right there, or thickness. The most abundant chemical inside the red blood cell is hemoglobin. So 1/3 of the volume of a red blood cell is nothing but hemoglobin molecules. And the role of hemoglobin, the biggest role, is that's where oxygen binds. So hemoglobin is the part that carries oxygen. Okay? And so hemoglobin, another thing about hemoglobin I did not write down, but I'd like you to know, it's a red pigment. And so that's what gives the red blood cells their color. Okay? So even if you put a drop of blood on a slide and you don't put a stain on it, it's still, the red cells are still going to look red because of the pigment hemoglobin. Okay? And when oxygen is attached to the hemoglobin, it's a bright red. When oxygen is not attached to the hemoglobin, it's more of a maroon or a dull, dark red color. Okay? So you have oxyhemoglobin, you have deoxyhemoglobin. Okay? One very interesting feature of a red blood cell is that it does not have a nucleus. So that lacking nuclei, that you could call that anucleated. The prefix a means not or without, right? Anucleated. Okay. So because they don't have a nucleus, they can't undergo cell division. So you got to be making new ones from the bone marrow all the time because they can't divide to produce other cells. Okay. They also don't have any mitochondria. So obviously they can't go through aerobic cellular respiration, can they? They do generate ATP, but it's just through the anaerobic phase of glycolysis. So they rely just on glycolysis for ATP. But that's all okay because they don't live that long. The lifespan, I should have put this in your notes, sorry. The lifespan of a red blood cell is about 120 days. So about 4 months. So they're made, they live 4 months, they die. Okay? Okay, so here's some other views of them with an electron microscope, a scanning electron microscope on the left in color, you see that? And then on the right at the top is showing you some with the transmission electron microscope. That's a sideways view of a red blood cell right there. And then at the bottom, this is a light microscope. This is showing you a blood capillary that has some red blood cells lined up. They pretty much have to line up in your blood capillaries. Many of the blood capillaries are just barely 7.5 microns in diameter, if that. So these cells have to be somewhat elastic to squeeze through some of your blood capillaries. Okay. Now, if we look at the red cell count, like how many you have in you, it looks like, okay, I got 4 to 6 million, depending on if I'm male, female, or a child, 4 to 6 million red blood cells. That's not true. It's 4 to 6 million per microliter. Do you know what a microliter is? It's a cubic millimeter. A microliter, this is microliter. That's the same thing as a cubic millimeter. Can you imagine a cube that is a millimeter on each edge? And you've got 4 to 6 million red blood cells within that tiny little bit of space. That's much smaller than just the typical drop, isn't it? Uh-huh. And so if you have 1,000 cubic millimeters, how many cubic millimeters are in a liter? I need to go sit down and figure that one out. It's more than 1,000, isn't it? Anyway, and then you've got 5 liters of blood in you. You see how you could figure out how many zeros to put on the end of the number of the red blood cells in you? It's an awful lot. But this count, it varies. You don't have to know the details on the numbers. Just know basically 4 to 6 million per cubic millimeter. Okay? 4 to 6 million per cubic millimeter. Those are the normal ranges. And your particular number, your red blood cell count, helps in diagnosis of a lot of diseases. Like a person who has leukemias, their red cell count is going to be less than normal. Their white cell count will be elevated, but their red cell count will be less. If you have fewer red blood cells, you're not going to get as much oxygen to your cells. So that's referred to as your oxygen carrying capacity. The more red blood cells you have, the higher the oxygen carrying capacity it is. Okay? All right, so that's some terminology you see medically dealing with red cells. Okay? All right. Now, if you have however many zeros would be at the end, gazillions of red blood cells in you, but they don't live but about 4 months, do you see how you've got to constantly be making a whole lot more every single day? All right, and the process of making red blood cells is called erythropoiesis. Erythropoiesis. So that is one of the branches of hematopoiesis. It's just the red cell course of events is called erythropoiesis. It occurs in the red bone marrow, and it occurs anytime your blood oxygen drops. Because whenever your blood oxygen drops, your kidneys and your liver produce a hormone called erythropoietin. I promised you in chapter 13 that we wouldn't be finished with hormones when we finish the endocrine system chapter. We're going to see another hormone in almost every other, if not every, unit from here on out, at least 1 or 2 hormones. But this one for this chapter is called erythropoietin. That's a hormone. Make sure you know that's a hormone. It's H. Hormone. Sometimes you hear it referred to as EPO. That's erythropoietin. Where is it produced? In the liver and the kidneys. Okay? Those are great places to produce it because the blood's filtering through both of those organs many times every day. Okay, so it cycles through this. So that's a good place for cells to figure out if there's enough red blood cells or not. Okay, if they're not, then they produce erythropoietin, and erythropoietin in turn activates a hemocytoblast to go through initially the myeloid line, and then from there to go through the red blood cell line. Okay, and so ultimately you have cells formed in the bone marrow called erythroblast. Remember, a blast is a cell that makes something. Erythroblast, that sets the fate. Okay, now it's going to become a red blood cell. And then there's another stage in there called a normoblast. But ultimately, you have a little cell called a reticulocyte. Sometimes we see those actually circulating in the bone marrow, but that is a little immature red blood cell. But it gives rise to the mature erythrocyte. So just in a few days after erythropoietin is released, you can have lots more red blood cells produced at your bone marrow. Okay, so I should have put this slide. Look back at this slide. Here you can see some of those cells, except this is so fuzzy it's hard to tell. Is this the erythroblast, the early erythroblast? All right, but notice as it gets from this stage, erythroblast, to the reticulocyte, what is it losing? A nucleus. And what is it gaining? If you were in second grade and you stood real far away from this, what is the difference in this cell and that cell? Hemoglobin, or the color is the difference. So it's gaining hemoglobin during this process. So if you this is for you to just think about it. It's not on the test. To me, it's really interesting. Do you see how initially there's a nucleus? And don't you need a nucleus to code for protein production? Yes, because there's a gene on that in the DNA, right? And there's a gene for hemoglobin. And evidently, from this point to this point, that gene is really being transcribed into messenger RNA that goes to the ribosome, where a lot of hemoglobin is made. And then once you have enough of that, you don't have the nucleus anymore. You may have some remnants of some organelles in there. Sometimes it looks a little speckled, but that's called a reticulocyte, a little immature red blood cell. So when you look at blood under the microscope, as we'll do in lab in a few days, if you see a red blood cell that has tiny little dots, that's a very early one. Sometimes they move on out. That's a reticulocyte. It moves on out into the bloodstream, and so you may see it in circulating blood. But mostly what you're seeing is the mature erythrocyte. So you don't need to describe that event like I did, but you need to know the sequence of those cells, getting from the hemocytoblast under the influence of erythropoietin to the erythrocyte. So hemocytoblast, erythroblast. I should have put myeloid stem cell, but erythroblast, reticulocyte, and then erythrocyte. Okay? Y'all good with that? Okay. All right. There are some things you need in your diet that affect red blood cell formation. Two of those are vitamins B12 and folic acid. Those are some of your B complex vitamins. They're necessary in all cells whenever they're dividing, because you have to have DNA replication when cells divide, right? And without these two vitamins, you won't have enough DNA production. All right, and even though the mature red blood cell does not have DNA, the precursor cells do. And because you're making millions and millions of those every day, you've got to have adequate amounts of vitamin B12 and folic acid. These are some vitamins that are very abundant in pregnancy vitamins. So they've, in the last 20 years or so, they've really noticed how folic acid deficiencies are detrimental to a developing baby. And it's because that developing baby, that is needing to produce lots of red blood cells because that growth rate is so fast. Iron is another a mineral that is needed in your diet because it's one of the components of hemoglobin. So in fact, it's the part of hemoglobin that oxygen attaches to. Okay. Look at this table right here, at this thing called intrinsic factor. All right, intrinsic, you don't have to write this down, but intrinsic factor is an enzyme, a chemical that's produced at your small intestine, and without it, doesn't matter how much vitamin B12 is in the food or vitamins you take, you can't absorb the vitamin B12. Some people genetically do not produce enough of that intrinsic factor. Therefore, they have a severe vitamin B12 deficiency. They can't make enough red blood cells, and it can be devastating. It's a type of anemia. You may have heard of it. It's not real common. It's called pernicious anemia. It's very severe. It can be fatal. So children who are born with that have to take intrinsic factor the rest of their lives so that they won't suffer that. But at least they can take it. So that's good. Okay. All right. What is the name of red blood cell formation? Erythropoiesis. Okay. What is the name of the hormone that causes it to occur? Erythropoietin. Where is that hormone produced? Liver and kidneys. When it's produced, tell me a sequence of cells that develops. From the hemocytoblast, erythroblast, reticulocyte, erythrocyte. Okay, and it happens a gazillion times every day, right? Okay. How long do those red blood cells live? 120 days. Therefore, every day you got a whole bunch of them dying, don't you? Okay, as your red blood cells get older, they become pretty fragile, um, and they can't produce new proteins because they don't have any DNA, and they don't produce tons of energy because they don't have any mitochondria. So they get fragile and they get damaged. So you have these wonderful cells in the body that are phagocytes, and they come engulf them and they eat them up like Pac-Man eats them up. And so you have got to have a way to take care of these old, broken down, destroyed red blood cells because you don't want them to just accumulate in your body. They've got to be processed. Parts of them are recycled, parts are not, and you get rid of it. So we're going to look specifically at what happens to the hemoglobin because there's a whole bunch of hemoglobin in a red blood cell, a third of the volume of a red blood cell's hemoglobin. And we're going to look at the breakdown of hemoglobin as these erythrocytes are destroyed. So that mostly occurs at your liver, also the spleen, but especially the liver. Especially the liver is where the breakdown occurs. The making occurs at the bone marrow, red marrow. The breakdown at the spleen and the liver. Okay. All right. And so this is what happens. I'll come back to that slide. Okay, look at this. What you're seeing on the left in the purple is a simplified view of the hemoglobin molecule. And do you remember all proteins are made up of a chain of amino acids called a polypeptide that takes on a three-dimensional shape, right? Sometimes a pretty complex three-dimensional shape. Some proteins are made up of more than one chain of polypeptides, and hemoglobin is one of those. Hemoglobin is actually made up of 4 chains. So do you see 1, 2, 3, 4 chains here? Or 4 up there. 4 chains? And in the middle of each is this little thing that looks like Saturn, doesn't it? Okay, that represents, that's what we call a heme, H-E-M-E unit, which is what you're seeing right here. That's a heme unit. And it's in the middle of each hemoglobin. And that's the part that the oxygen binds to. Do you see how in the middle of the heme there's Fe? Do y'all know what Fe stands for? Iron. Okay? And that's the part where oxygen is bound. So if each hemoglobin molecule has 4 heme units, that means it can hold up to 4 oxygens. Okay? All right. So when the hemoglobin is broken down at your liver, one of the first things that happens to it is that the heme units are pulled off, and then all you're left with are these polypeptides that are processed like any other protein is. They're broken down maybe into amino acids. You may reuse those amino acids, or they may be broken down further for other things. All right, but we're going to look at what happens to the heme unit. Okay, what color is hemoglobin? Red, and the heme unit is the red part of it. And only when iron is attached to it, the iron makes it red. Okay? But the first thing that happens to that heme unit when it starts to be broken down is that the iron is pulled off of it. And so now it becomes another color. See, here's the iron over here. It's taken off. And this is green. It's not red, but it's green. And it's another pigment called biliverdin. V-E-R-D in other languages refers to green, doesn't it? Like French and Spanish. Okay? Verde. But anyway, biliverdin is a green pigment, and it's the part of this entire molecule that you're going to get rid of. It is a waste product of hemoglobin breakdown. You might keep the iron. That iron can go back to the bone marrow, or it can be stored in the liver to be reused one day to make new red blood cells at the bone marrow. But the biliverdin you've got to excrete. And when it's made there at the liver, the place it's going to go is the small intestine. And it becomes incorporated into the fluid of the liver called bile. You may have heard of bile. And bile is a very important fluid that your liver produces. It's kind of a waste product of some of the chemical processing that occurs in the liver. And it goes into the small intestine and then it's just passed out with your feces. There's some digestive components to it that we'll look at when we do digestive system. But the biliverdin can further be broken down into a yellow pigment that you probably have heard of before called bilirubin. Now your book may say orange yellow, but it's really more yellowish than orange. Bilirubin. So biliverdin and bilirubin are so colorful that that's what gives bile the color of that yellowish green, especially the green. But the end course of this is the bilirubin. The biliverdin is converted to the bilirubin. But those are the waste products, and they become components of bile. Okay. Sometimes a person's liver doesn't function well enough to remove all of that and put it in bile, so it backs up into that person's tissues and can cause pale tissues to look yellow, or any tissue that is extremely vascular to look yellow, like the whites of your eyes. Your skin takes on a yellowish cast. Your gums take on a yellowish cast. What is that condition of the yellowness? Jaundice. You hear about jaundice. Somebody who has liver cancer could experience jaundice. Cirrhosis of the liver. Hepatitis, which is one of the viral infections of the liver. Sometimes a newborn has jaundice. Not when they're born, but a day or two afterwards they start getting it. And they have it for several days or maybe even weeks. And there's nothing, well, it's something wrong, but it may not be detrimental. Sometimes because their growth rate is so fast, their liver just has a hard time initially catching up. And the biliverdin and bilirubin especially backs up. So usually that corrects itself over time. But in the meantime, the nurses and doctors will tell the parents to put the baby in a crib, let the crib be in indirect light for an hour or two every day. The sunlight tends to break down the bilirubin, but it usually corrects itself. Okay. So here you can see that in the bone marrow, if you follow, let's do the 1, 2, 3, 4. You absorb lots of nutrients from your bloodstream, and those are in turn transported to your red bone marrow where you're making blood cells, and then as they get out in the circulating plasma, they live about 120 days. Then they are taken up by the liver and they're destroyed by macrophages. The hemoglobin is pulled out and converted to biliverdin, then bilirubin, which is excreted in the bile. And the bile goes into the small intestine where it's passed out with feces. Okay, so lot to know about red blood cells, isn't it? A lot to know. Spend a lot of time studying all of that. Okay, what's next? Okay, now we're going to look at the white blood cells. So we've looked at everything along this line. But now we're going to look at your white blood cells. Do you notice? Here's the circulating in the blood, the circulating white blood cells. Okay. It's kind of interesting. One of these can get out of the bloodstream to become a macrophage. And then this one gets out to become some other cells that we'll talk about it in another chapter. But anyway, but these are the white blood cells that I've circled. Okay. And do you see that they're in two groups, granulocytes and agranulocytes? Okay. All together, they're called leukocytes. Leuko means no color. They don't have pigment in them. Now, when you look at them under the microscope, they can be very colorful, but that's just because of the stain that's put on them. In their natural state, they don't have a color. But they also develop from those same hemocytoblasts in response to different hormones, and thank goodness you're not going to have to name any of those. Okay? But there's different kinds. We're going to look at 5 different types, but they can be lumped into 2 groups, either granulocytes or agranulocytes. So one of them has cytoplasmic granules, one type does not. Okay? A means without. Okay? And so when we look at the granulocytes, they tend to have what we call a lobed nucleus. So if there's the cell membrane, instead of the nucleus being a nice little round nucleus, it may look real lumpy. It may have 2 lobes, it may have 5 or 6 lobes in it. And sometimes just the way it's positioned, and when you're looking at it in 2 dimensions, it looks like they may be separate nuclei, but it's all connected. But they could have a lobed nucleus, and then they have little granules in the cytoplasm of varying pH. And with the stain that is used for blood slides, the stain turns a different color in response to a different pH. And so we'll look at 3 different kinds of granulocytes. One of them has neutral granules, neutral pH, so it stains a particular way. They're called neutrophils. One has acidic granules. You don't have to know which are neutral or acidic or basic, but the eosinophils their granules are acidic and they stain more pink and red. And then the basophils have basic granules which stain a dark bluish black color. So that's a big way you can distinguish between them. They don't live very, very long. You typically are making these. They're being used for whatever process, and then they die. Okay. And their production is a lot like that of red blood cells based on hormones. Okay. I was going to say something. I can't remember what I was just going to say. Oh well, I'll think of it. All right, then your agranulocytes don't have cytoplasmic granules, and their nucleus takes up a big bulk of the area inside the cell. Some of them, the nucleus is humongous, and it has very little cytoplasm. Some of them, the nucleus is like, it's maybe kind of bean-shaped, or it may all look a little bit bilobed. But some of these live just a very short time. Some of these live years and years and years. Oh, I remember what I was going to say. If I was to ask you what's the function of a leukocyte in general, they individually do a lot of different things, but it all boils down to protecting you as body defense in one form or fashion. They work in body defense. So, okay. All right, the different kinds of agranulocytes are the monocytes and the lymphocytes. Okay, so we're going to start with the neutrophils, and I've got several slides that show these for you. What you're seeing down here are mostly like this and this. These are the red blood cells. And do you see how it looks lighter in the center on a lot of these? That's that indication that it's thin in the middle, that biconcave disc. But the white blood cell, like here's a white blood cell, it's a neutrophil. Here's another neutrophil. And see how it's got a highly lobed nucleus? It's by far the most abundant of all the white blood cells. In your lab class, you'll be looking at these again, and you'll have the opportunity to do a count of these, and you'll see that there's lots and lots of white blood cells. But you do need to know these percentages. Okay, so they're over half of them. They're the most active phagocytes of all of your white blood cells. So that's how they protect you, is that they act as phagocytes, especially if you have a bacterial infection. So if you're struggling with some type of bacterial infection, your neutrophil count's going to go higher than the 54 to 62%. Okay, because that's an indication that it's bacteria that you're fighting. So they're very, very, very good phagocytes for you. Okay, then look at this one. Compare these cells to these, especially the one on the right. It shows it a lot better. Look at the granules. See how they've stained red? And do you see that mostly it's a bilobed nucleus? All right, these are the eosinophils. And they also are phagocytic. They also elevate during allergic reactions. But as far as phagocytosis concerns, let's say if you're battling some type of parasitic infection, like Giardia, which is a protozoan parasite, or tapeworm infection, any of these parasitic infections, eosinophil count's going to go up. Look at the percentage. 1 to 3% of all your white blood cells are these. So they're not nearly as abundant as the neutrophils are. Okay. All right, then the basophils, look at their granules. Their granules stain so darkly, especially on that left side, you hardly even see the nucleus, do you? Right? Okay. They have dark blue staining granules, and these are not phagocytic, but they release some chemicals that are very important for you. One's called heparin, one is called histamine. They both start with an H, but they don't have anything to do with each other. Heparin is a real powerful anticoagulant, which prevents blood clotting. So if you're producing a lot of that, you may do that when you need more blood flow into an area. So this basically thins your blood. Some people don't produce enough of that naturally, so their blood tends to clot too much. Histamine is a chemical we've talked about possibly in the past that promotes the inflammatory response. Do you remember, maybe you remember this, in A&P1 when you studied connective tissues? One type of connective tissue cell was called a mast cell. A mast cell produced heparin and histamine. But it's like a basophil, but it just sits out in your tissues instead of in your bloodstream. Okay, but it does the same thing. All right, as far as the abundance of these, probably because we have mast cells, you don't need quite as many of these. Less than 1%. So when I see these in lab, I get really excited because it's less than 1% of all of those white blood cells. Okay. All right, now neutrophils, eosinophils, and basophils. Those are your granulocytes. And they all tend to be about the same size, like 1.5 times, or about, I don't know if it's that number. But compared to the red blood cell, you can see their size. They're bigger than red blood cells, a little more than twice as big, I think. But anyway, the agranulocytes are different in their sizes. Okay. The first one we're going to look at is a monocyte. It's the largest of all the white blood cells. So compare this pictures of these monocytes to the white blood cell to some of those granulocytes to the white blood cell. These are the largest white blood cells. And I know that when you look at these slides, it looks like there's little granules in the cytoplasm, but they're not nearly evident like in the granulocytes, are they? And actually, through the microscope, I like to describe it as a smooth blue cytoplasm. It stains sort of a smooth blue, a light blue. I know that this picture doesn't look blue at all. These are just some bizarre pictures from an older book. The one in your book is much better. It's like the one on the right. But do you see how the nucleus takes up a bulk of the inside of the cell? And that it's overall somewhat bean, B-E-A-N, bean shaped, or kind of sometimes it's more horseshoe shaped. Okay, 3 to 9%. These are also phagocytic. So because they are so large, they can phagocytize things bigger than bacterial cells. So these actually phagocytize some of your old worn out cells. And it's interesting to me when we talk about macrophages or big humongous phagocytes, what a macrophage is a monocyte that has gotten out of the bloodstream to go out into your tissues. Okay. And then the lymphocytes are the smallest of the white blood cells. You know, it's not a whole lot bigger than a red blood cell, is it? But do you notice how it has just a little thin rim of cytoplasm right there? But the bulk of it is just this humongous nucleus. Okay. Okay, that's a lymphocyte. There are a couple of different kinds of lymphocytes, but they pretty much look the same initially through the microscope. But these, the big significance of these is that those are involved in immunity, and that means they can produce chemicals like antibodies, which we'll talk about a lot in another unit. Okay. So they're the second most abundant white blood cell. See, 25 to 33% of all your white blood cells are these. Okay. All right. So here, right here at the bottom, you can see those 5 kinds, putting the 2 lymphocytes together as one. Okay. So you see, here's the eosinophil. See how it's got the red colored granules? Not naturally, but in the stained state under a microscope. And then there's a basophil with the dark granules. The neutrophil, the granules don't really show up a lot in the neutrophils, but you're going to see so many neutrophils when you look through the microscope at blood slides. You'll learn what a neutrophil looks like. Okay. And then the monocyte, look how big it is. And then the 2 lymphocytes. Well, you just lump them together as one lymphocyte. They have a big nucleus, very little cytoplasm. Okay. All right. And then here's, I pulled this from the web. You can see it again. I know colors, depends on your stain. But colors look a little bit different, don't they? And if you compare these, you can see that this one, the granules don't stain up a lot. But here they're kind of pinkish red. Here they're super dark. I think a basophil just looks sort of freckled. I just think of freckles when I see a basophil. And then the lymphocyte has very little cytoplasm. And then the monocyte is just big. And it has a kind of that smooth blue cytoplasm like the lymphocyte does, instead of granular. And it has a bean-shaped nucleus, or in this case, horseshoe-shaped. All right, and then there's the same pictures I had put before. These little things, by the way, right there, those are some platelets, which we'll look at in a minute. Okay. And here's some platelets right there. Okay. All right. And okay, now you've heard of leukemia. I am not going to test you on leukemia, but you probably all know of someone who's been afflicted with that. But it is a cancer in your bone marrow of the white blood cell, the ones that are going to become the white blood cells. There's different kinds. Some of them can progress real quickly, and you get symptoms just all of a sudden. Some, you kind of slowly get these symptoms of fatigue and pain, and it's just kind of flu-like, and it just lasts and lasts and doesn't go away. That's the more chronic kind. But anyway, some of them are of the lymphocytes, and some are the myeloid type, which particularly affects the granulocytes. And I think of the granulocytes, it's more the neutrophil line. But anyway, here you can see some symptoms of it and treatments. One of the final treatments, last resort treatment, is the bone marrow transplant. But what they do with that is the chemo drugs, or chemo targets mitotic cells. All right, and because in leukemia these cells are they're dividing too much, the chemotherapy is destroying them. So they pretty much, if they give massive doses of that, it's destroying the person's bone marrow. And so when they don't have that bone marrow, they have to be put in an isolation unit in a hospital. And then someone else the donor is given drugs to increase all of their blood cell formation. And they may have some flu-like symptoms initially, but it's just to get more bone marrow in there. And then they'll pull some of it and put it in the patient whose bone marrow has been destroyed. through all the chemo drugs, hoping that person will take it well, and then he'll have maybe even a whole new blood type, because he's got all new stem cells in his bone marrow that are not the cancerous kind. So they have been very successful. It's a very touch and go process, but when it works, it's really good. And okay. Now, we talked a little bit about histamine being released by basophils, but it's also released by mast cells and then just damaged cells. But what that histamine does is set off inflammation. And so one of the first things that happens during inflammation is that blood vessels in that area dilate, and that means more blood can flow to the area. Then the blood vessels are more permeable, and that means more fluids leak out of the bloodstream, and the tissue swells. And that swelling could be enough to contain the pathogen, so it doesn't go anywhere else. So it results in red, swollen tissues, and that all of that prevents that spread of microorganisms. Because more blood flows to the area, it gets warm, and that heat might denature some of the proteins on the pathogens. Phagocytes are attracted to the area, and this is where some of the white blood cells come in. Some of these phagocytes exhibit a process called positive chemotaxis. Taxi means movement, doesn't it? Taxis. Chemotaxis means movement in response to a chemical. Positive chemotaxis means movement toward the chemical. Okay, and a lot of phagocytes exhibit that process of positive chemotaxis. Damaged cells release chemicals that attract phagocytes. Okay, so phagocytes like neutrophils come there, and they can phagocytize the pathogen. The combination of the fluid release and the white blood cells and the pathogen. I know it's gross, but that's what pus is. It's the combination of all of that. Okay. So here you see a splinter causing a little infection. So the little green thing would be, these are bacteria, and you can see some neutrophils coming in to engulf that. Okay, I mentioned that just overall, the function of white blood cells is body defense, but these are some of the ways that they can do that. I've already talked about phagocytosis is one way. I've already talked about inflammation is another way. I've talked about this positive chemotaxis, which is a combination of all of that. I've talked about antibody production. Some of the white blood cells produce antibodies. Another feature of white blood cells that's pretty cool is that they can exhibit what we call diapedesis. Diapedesis means that this cell, not all white blood cells do this, but some of them do, but this cell can actually move out of the bloodstream. The blood vessel wall, typically it's only chemicals that cross it, like water, like calcium, oxygen can cross it. Not a whole cell, but some white blood cells can do that to get to your tissues. Cancer cells, many cancer cells can undergo diapedesis. That's why it's so easily spread to other places of the body. Yes, I said a macrophage is a monocyte that went through diapedesis to get out into the tissues. Yeah, but your neutrophils can do that too. Okay. All right, and if we talked about the red cell count, the total red cell count between 4 and 6 million per cubic millimeter of blood. Okay, the white cell count is not nearly that much. The total white cell count here, see, it's not anywhere near in the millions per cubic millimeter. So you're not going to see near as many white blood cells as you do red. But your total, this is your total white cell count, the total one, the total. The total white cell count is, it ranges. The range varies with whether you're man, woman, or child. And it's going to vary based on if your body's trying to fight anything at any given moment. Okay, but if you consistently, if you have an elevated white blood cell count, That condition is called leukocytosis. Okay, more than the 10,000. If you have a depleted white cell count, that's called leukopenia. Okay, and usually that's a temporary thing. Okay, now a differential white blood cell count. This is very important for you to know. Okay, this one is the total white blood cell count. And how's that different from a differential white blood cell count? That doesn't just tell you the total number of white blood cells in a cubic millimeter. It tells you the percentage of each kind of white blood cell in your blood. So when I gave you those percentages, like for neutrophils, eosinophils, those were differential counts. And that's an important diagnostic on blood. Let's say somebody has the symptoms of pneumonia. Right? Some of you may realize that pneumonia can be caused by certain viruses, but pneumonia symptoms can also be caused by bacteria. And you would treat them with different drugs, wouldn't you? But let's say they don't know which kind you have. But if they do a differential white blood cell count and they find that the neutrophil count is 70%, that's higher than the norm, isn't it? In the normal range. So that's an indication that you're fighting bacteria because neutrophils engulf bacteria. So then they treat you with antibiotics, which would not be effective if it was a viral type of pneumonia. Antibiotics are effective against bacterial infections. Okay? So in a medical lab setting, these differential counts are very, very important. And that's why I want you to memorize the percentages. Those percentages are the norms for differential counts. Okay? All right. All right, and then the last formed element would be the platelets, and they're called thrombocytes. And they're just little fragments of cells that develop in the bone marrow. So if we look at that bone marrow slide again, here you see little platelets. All these little dots that you see are platelets. And if we look at this bone marrow slide, and if we follow this course, do you see if the hemocytoblast goes through this line, it becomes this humongous cell with a big nucleus. That's why it's called a megakaryocyte. Karyo refers to nucleus. This is a cell with a huge nucleus. It's a huge cell with a huge nucleus, and that breaks up to form the platelets that then circulate out in the bloodstream. Okay? Okay, and so I'm backing up. Platelets, of course, they lack a nucleus because they're not whole cells, but the red blood cell is considered a whole cell because it initially had one. Platelets never had one because they came from broken down pieces of cells. Okay, and we're going to see their role next time as we look at some of the mechanisms that occur with blood. And one of those mechanisms is called hemostasis, and that helps prevent bleeding when you've got some damaged blood vessels. And platelets help with that. They release a chemical called serotonin, and serotonin triggers blood vessels to constrict. Okay, we'll look at that process next week. Just for right now, know that platelets release serotonin. The chemical they release is called serotonin. Okay, but what serotonin does is actually cause smooth muscles to contract, and so that triggers some of these tiny blood vessels to close up, which is important if there's a tear in the wall of that blood vessel. It prevents it from bleeding out. Okay. Okay. And there's your platelets, all those little specks. Those are platelets. And so here's an overview of what we've covered today of the solid components of your blood. So that would be a good study tool. All right, next time we'll look at blood plasma, and then we'll look at all the functions that occur in the blood.